Okay, welcome back to the lecture. We're going to continue talking with certain effects of training on muscle fiber types. We're going to break those into oxidative and glycolytic fibers. So muscle fibers that are frequently active respond by increasing their performance capabilities. The changes that occur are going to be different for different muscle fiber types. So let's talk about oxidative fibers first. With training, we can increase myoglobin content, and if you remember, myoglobin is a protein that enhances oxygen delivery within the muscle cell. Increase the number of mitochondria within the cell, so that enhances oxidative capabilities. We can increase the number of capillaries serving the muscle, so that enhances blood flow and exchange of nutrients to and from the muscle. We can increase oxidative enzymes, so that enhances our oxidative metabolism pathways. And all of these changes involve very little changes in muscle mass itself. So you will not see too much hypertrophy in type 1 muscle fibers, which are considered our oxidative fibers. Now, switching gears down to our glycolytic fibers, we can, with training, increase the number of myofibrils, thus increasing the number of contractile proteins. So we can increase the number of actin-myosin. So when we increase the number of actin-myosin proteins, myofilaments, what we can do is we can enhance contractile force, and that's how we gain strength and become stronger as well. We can increase our glycogen stores, so we can store more glycogen and enhance anaerobic capacities with glycolytic pathway. Addition of more collagen and other connective tissues that must sustain the increased tension. So we can actually lay down more collagen as we get stronger to protect our tissues a bit more. All of these changes necessitate an increase in fiber diameter and thus an increase in muscle mass. So our glycolytic fibers, again, are going to be the ones that respond to hypertrophy. We will talk about that a little bit more in our next lecture, but the glycolytic fibers are going to be the ones that's where you gain muscle mass, is where you gain the girth and the size, because we're increasing the number of myofibrils, increasing the number of actin-myosin filaments, and when we talk about hypertrophy next class, we will be talking about how sometimes satellite cells can incorporate into muscle cells as well. All right, let's continue on with muscle fatigue. What is muscle fatigue? This is the use-dependent decrease in the ability of muscles to generate force. So the rapid fatigue and high-intensity exercise apparently results from operating muscles in non-oxidative conditions. So this is, for example, if we're lifting weights or if we are sprinting. That's why we can't sprint or lift weights indefinitely. We have to rest and recover for a number of reasons. High-intensity exercise causes an increase in lactate and hydrogen in the blood, which causes a drop in the pH, right? So we talked about that with anaerobic metabolism. If we are going through the glycolytic pathway very fast, we produce a lot of lactate and we produce a lot of hydrogen, which ultimately that increase in hydrogen is going to cause that drop in pH. So high-intensity exercise and a decrease in pH with exercise alters muscle physiology in the following ways. Enzyme activity it affects our contractile machinery. It depletes high-energy phosphate compounds. It causes an alteration in the ionic gradients across the cell membrane and the transverse tubules. It interferes with the ability of the sarcoplasmic reticulum to control intracellular calcium concentrations. And also, a big one is that hydrogen competes with calcium for the binding sites on troponin. So that's very important, right? Because last we spoke about muscle contraction. We need calcium to bind to that third molecular subunit of troponin, which causes the conformational shift of the tropomyosin strands out of the way in order for the myosin to grab onto the actin. Now, if hydrogen is competing for calcium for that spot on troponin, that means there's less spots for calcium to occupy. So then we're not going to have as many cross-bridge cycling going on. We're not going to have as much myosin grabbing onto the actin, and that will become an issue. And that's where we will see a decrease in force, contractile force, and output. All right, so let's talk a little bit about slow fatigue and what that is. The slow fatigue associated with prolonged submaximal exercise coincides with the depletion of the glycogen stores of the liver and of our working muscles. When this occurs, the body must rely on what? On fat as the only energy source. Since energy production from fat occurs at half the rate of the energy production from glucose, there is a substantial decrease in muscular performance. So that makes sense. So if we are doing prolonged exercise and we really only can at this point rely on fat for our energy source, we know that fat takes a lot longer to break down and to produce all that ATP. Don't get me wrong, we have a huge amount of ATP produced from one free fatty acid. However, it takes much longer than when we get ATP from glucose. So if we're only relying on fat, you will see a decrease in muscle performance since it is taking us so long to get that energy that we need because we have to rely on our fat stores. Okay, so now we are going to shift gears and start talking a little bit about motor control. And before we get right into the brass tacks of motor control, we're going to go over some basics again about the spinal cord. Hopefully this should be a review for most of you. So the spinal cord. It's a cylinder made up of gray and white matter, right? Where is this located? It's located in the upper 2/3 of the vertebral canal surrounded by a bony column. This is our primary central processing center. So ultimately it's a relay station. It extends from the foramen magnum of the base of our brain and extends and ends at the cone-shaped termination called our conus medullaris. So do you remember from gross anatomy, our foramen magnum comes out of the base of our skull. The conus medullaris goes to the first lumbar vertebrae, and there's a non-neural set of filaments that continues towards the coccygeal region, and it gives a point of attachment for that end of the spinal cord, known as the filum terminale. Filum terminale, those are hair-like filaments that come off the conus medullaris that give us our southernmost attachment in our coccyx region. Okay, from top to bottom, the spinal cord is covered with 3 pieces of connective tissue called meninges. Our innermost layer, which is closest to the spinal cord itself, is called the pia mater. This is highly vascularized. Our second layer is a non-vascular piece of connective tissue called the arachnoid. And in between the arachnoid and the pia mater, there is a subarachnoid space. That subarachnoid space is filled with the cerebral spinal fluid. And floating in this fluid, there are actually some blood vessels there. The third piece of connective tissue is the most exterior, or the outermost, and it's called the dura mater. This is the most fibrous of all 3. So if we go to our picture here, again, this should be a review. We have our pia mater here, and we have our arachnoid mater here, and then our dura mater. So these are 3 layers of connective tissue surrounding our spinal cord right here. And again, the pia mater is highly vascularized here, and so forth, and the arachnoid is not. So if we quickly talk about the blood supply to the spinal cord, it gets its blood supply mainly off of 4 main branches, which are vertebral artery, cervical artery, thoracic artery, and our lumbar artery. We also have veins that go in opposite direction to take blood away. So we have our vertebral vein, cervical vein, thoracic vein, and lumbar vein. And we know we have a bunch of different branches that feed blood to the spinal cord. So we're not going to go get into the nitty-gritty of all the blood supply to the spinal cord and so forth, because there's the anterior spinal artery, posterior spinal artery, spinal branch, all these other good things. But for the most part, those are the 4 major branches which all these other branches come off of. Okay, so let's talk about spinal root and peripheral nerves. So when nerves come off the spinal cord, they actually come off in a series of dorsal and ventral rootlets, which join together to make 31 pair of mixed spinal nerves. The dorsal rootlet will have a bump on it, and the dorsal rootlet will combine with the ventral rootlet and make the mixed spinal nerve. As you go down the spinal cord, you will see 2 enlargements. You will see a cervical enlargement and a lumbosacral enlargement. These enlargements are clusters of cell bodies and are necessary because that's where the nerves come off to innervate the upper and the lower extremities. Okay, so you can see right here we have our cervical enlargement. This contains all of our neurons that innervate the upper limbs, and then we have our lumbosacral enlargement, or our lumbar enlargement, sometimes you will just see, contains the neurons that innervate our lower limbs. Okay, so let's talk about these rootlets. A dorsal rootlet is considered sensory. A ventral rootlet is considered motor. So we have general somatic afferent nerves and general visceral afferent nerves. We have general somatic efferent nerves and general visceral efferent nerves. General meaning distributed throughout the body. Somatic means going or coming from skeletal muscle. And visceral means going to or coming from glands and organs. Okay, so the major difference between sensory nerves and motor nerves is that the sensory nerve has a dorsal root ganglion. You can see right over here, right, coming off the spinal cord. This contains the cell bodies for the afferent nerves, so for our sensory nerves. The motor nerves don't have one because those cell bodies are actually located in the spinal cord itself, in the gray matter, more specifically, within the gray matter here. Right, so the ventral root, which is our motor pathway over here, we don't see a bump because they do not contain the cell bodies. Those cell bodies are actually in here. And this is where the cell bodies are of our dorsal root, our sensory nerves. Okay, so let's talk about sensory nerves and how they're classified by conduction velocities. So we can classify them this way as well. And they're considered groups 1 through 4. So if you ever read group 3 afferents or group 2 afferents, then you kind of know their function and you know what they're talking about. Okay, so let's start with group 1A and 1B afferents. Group 1A afferents are afferent nerves coming from muscle spindles. Now, what are muscle spindles sensitive to again? Muscle stretch. So muscle spindles monitor stretch in 2 ways. Violent stretch or a gradual stretch. Ballistic stretch monitors velocity of that stretch. 1B are sensory nerves, or sensory coming from Golgi tendon organs, right? What do Golgi tendon organs do again? Those monitor muscle tension. Golgi tendon organs are located where the tendons are attaches to the muscle fibers themselves. And Golgi tendon senses force, so too much force, you need the muscle to relax. So our group 2 sensory nerves, these monitor impulses coming from encapsulated receptors. So these actually will report back from the skin and from the joints. So touch, pressure, temperature, and joint movements, these are the afferents that respond to that. And conductivity is not as great as type 1s. Now, group 3 and 4 afferents are considered non-encapsulated receptors, and the thicker the axon, the faster the speed of the movement. So if it's myelinated, it equals, it's faster movement, and unmyelinated, it's slower. So ultimately, these are free nerve endings. Okay, so we're going to just quickly talk about motor nerves, the efferent portion. We have general somatic and general visceral. So again, somatic going to or from skeletal muscle, general visceral going to or from glands and organs. And we have 3 types of efferent fibers. Our alpha motor neurons, which goes to our motor end plates, which we talked about all last lecture. We have our gamma motor neurons. These innervate our intrafusal fibers on the ends of muscle spindles. So our muscle spindles are inside the fusiform capsule. So these intrafusal muscle fibers are actually skeletal muscle fibers that serve as specialized sensory organs, so proprioceptors, that detect the amount and rate of change in length of a muscle. And then lastly, we have our preganglionic autonomic neurons. These are first in relay of our autonomic signals. So we have preganglionic and postganglionic nerves, and there's a sympathetic trunk that actually runs up and down the aorta itself. Okay, so in the next video, we're going to continue on with the concept of motor control, and we're going to start talking about where the decisions are made in the brain, and we'll go from there.